some cool protein that you want to study, so you want to get cells to make it for you. First of all, how are you going to find the instructions for making them? And second of all, where are you going to put them? There are lots of different options when it comes to plasmid vectors, which are these circular pieces of DNA that we can stick the instructions for making a protein and stick this into bacteria and get bacteria to make a lot of it for us. They're also plasmid vectors for other sorts of cells expression. So today I want to talk more about these vectors and how we choose them, and also where we can find the instructions for making a protein we want to study.
Okay, so I have more background in other posts if people are interested. Today, I want to go really in depth into some of the features of various plasmids that you would want to keep in mind. And so just a quick background so we're all on the same page. We have the instructions for making a protein are going to be written in DNA in the form of genes. And this DNA, there's going to be a messenger RNA copy of it made. So an RNA copy of this gene gets made, and then this RNA copy, this messenger RNA, is used by these ribosomes, these protein-making complexes, to follow the instructions of the messenger RNA to make the corresponding protein. This messenger RNA is a little bit of a complication when it comes to the terminology that we use, because this messenger RNA is actually an edited form of the gene. The gene has these regions called introns, which have regulatory information that don't have the instructions for making the protein. So the instructions for making the protein are in the parts called exons, and the introns get removed in a process called splicing, and this gives you the mature messenger RNA. Let's talk about in some other posts. This can actually, you can splice in different ways, alternative splicing, and get different cDNA, different products, different messenger RNA products. When we want to stick the instructions into a vector, into this vehicle that's going to let us get it into cells, when we do this, we're going to want to put in DNA because this plasmid vector is going to be DNA. So we need to put in a DNA version of this RNA, and we call this a complementary DNA or a cDNA. So it's going to be the cDNA that we're putting in, not the actual gene, but we typically often just refer to it as the gene, but it is the cDNA. Sometimes you might see the term ORF, open reading frame. So this is also going to be a term that you might see, which refers to which frame that the reading is going to occur in, because of the different reading frames of the DNA. And so the open reading frame that you want is going to be the one that starts with so you have your ATG typically, or an AUG in the messenger RNA, and then the protein instructions. And so sometimes you might see ORFs in terms of the inserts that you're putting in. So the plasmid that you're putting into, we'll get way in depth into some of the features. But there are different things about the plasmid that it's going to have. It's going to have an origin of replication, and as we'll talk about, these can be high copy number or low copy number. This is where the DNA polymerase is going to latch on and make copies of this plasmid. So this plasmid is going to be hosted in the cell apart from the cell's own DNA. So the cell will have its own genomic DNA, as well as this plasmid that you're going to put into these cells. And both will need to get copied, and so it's going to have an origin of replication so that it can get copied. It's also going to have some sort of selection marker, such as an antibiotic resistance selection marker, and this way that only cells that have taken in the plasmid will be able to grow, and you can use this to select for them. We'll get more into each of these things in detail. In front of your gene, your insert, your cDNA, you're going to have a promoter. This is going to tell the RNA polymerase, so the one that is going to make this copy, this messenger RNA copy from the DNA, is going to tell it to make the copy. And then in bacteria, this is actually coupled. So when you have transcription happen, now you have translation happen concurrently, because the processes aren't separated like they are in our cells. So these are some different key features of plasmids, and then we'll look at how these can differ between different plasmids and how we might want to choose one or the other. If you're interested in how we actually go about getting the DNA, I'll go into more detail about how you can actually get the insert that you want to put in. There are depositories like Addgene and DNASU that we'll look at that actually host a lot of different plasmids with the instructions for making different proteins. So either ones that people have studied before and then they've like done papers on them and deposited them, or you can find ones that no one's really studied that much, but they have these giant libraries of all the cDNAs for these different proteins. And so we'll look at more of that later. Once we have one of those, we can then clone them into a different cell that we would like better. So even if we get a cDNA that comes in one plasmid, that doesn't mean that we're restricted to using it in that other plasmid. In fact, a lot of times you'll get a plasmid that's going to come in it's going to come in a vector that you don't want. It might even come in a vector that's totally incompatible with what you want. Maybe it's a mammalian cell expression vector and you want to express it in bacteria. And so you can then subclone it. So move it out of one clone into another. There are different methods for doing this, including restriction cloning, where you're basically cutting around the insert. I'll also show you these vectors often have restriction cut sites around them to make this easier, as well as PCR-based methods like SLIC, which is what I've typically used. There are also other like overhang-based methods, and sometimes the plasmids that things come in are like gateway plasmids or that sort of thing, where they're designed to have easier cloning between different plasmids. But traditionally, it's been restriction cloning until PCR-based methods really took over and that sort of thing. Okay, so when it comes to plasmids, it can be really overwhelming. So this is just from GenScript's website. They have this giant list of their vectors. And so you can see the ORF clone, so the open reading frame. And so I will warn you, though, that they're probably going to be a lot more expensive than if you were to find it, say, at DNASU or Addgene, and then just clone it into the vector that you want. And so hopefully this post will help you understand how you can do that more easily. But you'll see that there's going to be vectors for mammalian cells, bacteria cells, yeast cells, baculovirus cells, so like insect cell expression, as I've talked about before. And they're going to have all of these different features that we'll get into, things like tags, selection markers, different promoters. When you are looking for a plasmid in, say, Addgene or DNASU. Often these are going to be, sometimes these might be plasmids, especially with Addgene. There'll be plasmids that people have done a paper on, used in a paper. And so this is a paper that they actually use to express this protein, which can be a good thing because then you don't need to worry about cloning it into an expression, like a vector for your own expression, and because you know that it probably works and you don't have to optimize it too much. But if it's also one of the ones that's less studied and that sort of thing, they'll often be in some sort of just generic donor vector that we'll talk about. And so you'll see when you search for a different protein, you'll see a bunch of different things come up. And in various different plasmids, depending on how much people have studied this thing, they might have it in a lot of different plasmids, or if it's something that people haven't studied very much, then it's likely to just be in one or two plasmids, often just one. And it's just going to be a plasmid that's going to be good for cloning and not for expression. So let's talk more about the sort of differences between vectors that you might want to use for various purposes. So when it comes to comparing vector plasmids, here are some of the key things to look for. So remember, like, this looks really overwhelming. There's all of these different things. And even if we just look at PET vectors, which is the system of expression from this T7 promoter, and this is one of probably the most commonly used expression vector for bacterial expression. And you can see that even within this PET series, there's all these different things, but these really just differ in some really simple things that we're going to get into. And a lot of times, especially if you're using a PCR-based method, it doesn't really matter which of these your plasmid is in. Okay. So we're going to talk about these things now. So the copy number. So copy number refers to how many copies of the plasmid that the cell can host. So remember that the cell has its own genomic DNA, and then it's also holding copies of your plasmid. Some plasmids have what we call a high copy number, which means that there are lots of copies of them hosted in the cell, whereas others have a low copy number, which means there aren't very many. Often these low copy number plasmids are low copy number because they're under the control of the bacterial replication machinery and the bacterial replication proteins. So they're going to be dependent on those, and therefore they're going to only replicate like when the bacteria is, and so then they're going to have a lower copy number. And you might think that this is a bad thing, and it would be a bad thing if all you were caring about was cloning. So all of you were caring about was making lots and lots of copies of this DNA. Then it would be important to have a high copy number. But if then you subsequently want to get the cell to make protein from that, if you give them too many copies of the recipe, now they're going to maybe get sloppy, they're going to be rushing, they're going to run out of supplies and get depleted, and you'll have problems. You might even have like buildup of toxic proteins inside the cell. So this can be a bad thing. So typically a lower copy number would be better for protein expression, and a higher copy number would be better for just doing cloning and that sort of thing. If you want to make a lot of DNA, various things like that. So there are, this is going to depend on that origin of replication. So Addgene has some really great resources. They have this Plasmid 101 blog and guide that I highly recommend. Here you can see that they have a description of some of the common vectors and the various copy numbers. So some of the high copy number ones would be like your PUC vectors, your BlueScript, your pGEM. These have around 500 copies per cell. Whereas the ones that we use for expression, such as this PET vector, pGEX, PBR322, which is actually like the parent of these two, these are going to have a lower copy number, around 15 to 20. You can also see that some of these there's like these incompatibility groups. So sometimes you might want to put multiple plasmids inside of cells, and you need to make sure that their origins are compatible with one another. You don't want to put in ones that are in the same like incompatibility group. So if they're using like the same machinery, you don't want to put in two plasmids that are using that same machinery. Then they're going to be competing, they're going to be depleting the machinery, you're going to have problems. So you want to use ones from different groups. Okay, so that was the copy number. And now let's talk about the promoter usage. So when we talk about the promoter usage, now we're talking about this section. So you can see that in these pcDNA clones that were from mammalian cells, these are under this promoter called CMV. We're not going to talk about mammalian cell expression here, except to note that these plasmids, these pcDNA 3-point somethings, those are going to be mammalian and CMV. And so when you're looking in like Addgene or DNASU, when you see one of those, it's going to be a mammalian expression vector. But remember that you can always just subclone it out of that vector and into a vector for bacterial expression, etc. You might also see sometimes the vectors that are used are for like transient expression, some are for permanent expression, some are for various things, some have things added on like GFP. But remember, you can always subclone out and into whatever you want. Especially with PCR-based methods, this can be very simple, and you can do it from anything to anything. Okay, but for bacteria, now we're looking at, for our promoters, we're looking at things like T7 and LAC, and this is going to be T5, which uses the RNA, the E. coli RNA. These like PQE, these are going to use the E. coli RNA polymerase. There's also like this CAC promoter. So this is a hybrid between the TRP and LAC operons. It is capable of the inducible expression. So you add IPKG just like you would in your T7 and LAC ones. But in this case, it's going to use the bacteria's own RNA polymerase. And so it's a hybrid from these bacteria's own gene's own promoters. And so it's going to use the E. coli's own RNA polymerase, but it's going to have that IPTG inductibility. But because you don't need the T7, so you don't have to have special cells that have the T7, but that also means that you don't get that extra level of control. So you're going to get some more leakiness of the expression. So it's not good if you have something that has like toxicity to the cells. You want something with a tighter control. But yeah, so these PGEX1s have this TAC promoter, which I'll talk about more in a minute. For baculovirus expression here, you have like polyhedron or P10. You have other things for yeast. And so when it comes to this T7, this is often done in this inducible fashion so that you can induce the expression of T7. So what do I mean? Well, first of all, what is T7? T7 is this bacteriophage or phage. It's this virus that infects bacteria. And more on this in another post, but it's actually really cool in that it uses its own T7, it uses its own RNA polymerase. So the RNA polymerase, remember, is the thing that's going to make the mRNA copy from the DNA that the ribosomes can then make a protein from. T7 uses a different promoter than E. coli RNA polymerase. So the promoter is the part where the RNA polymerase is going to latch on and start making the DNA. The E. coli RNA polymerase is going to depend on a different promoter. And so the T7, it's only going to work on genes that have the T7 promoter. So if we put the T7 promoter in front of a gene that we want made, then we can get that gene made on demand. Or if we put the T7 promoter in front of our gene, we can get our gene made. And if we put it so that we are also in controlling the expression of T7, then we can control the expression of our protein. And this allows us to do this like inducible protein overexpression. So often when we have that T7 promoter, we also have the T7 actually provided from the bacteria's own DNA. There's integrated with this thing called like a DE3. When you see the words like DE3 inside of the cell name, this is going to refer to this lambda DE3. This lambda DE3 is going to be this like glycogen. It has the instructions for making T7, and it has a T7 under the control of this the LAC promoter. And so typically there's this protein called the LAC repressor that's going to sit on there and prevent the RNA polymerase from binding and making this making this LAC Z gene. So this is what actually happens inside of bacteria, like why they have this gene. But in the case of our systems, what we have is that we have this LAC promoter in front of the gene for making T7 polymerase. So bacteria use this so that they can control when they make the proteins that are needed to break down lactose. They don't want to make these proteins if they're not going to need them. And they're only going to need them if they don't have much glucose and if they have a lot of lactose. So when you have a lot of lactose, you get this glycosylation. So there's this little like change in the molecule that is going to give you this molecule called allolactose. Allolactose can bind to that LAC repressor and cause it to fall off. This de-represses things and then allows for the LAC-Z protein to get made, which can be used to in part of the process of metabolizing and breaking down this lactose that's in the environment to use it for energy. We can use a molecule called IPTG to mimic this de-repression, or we can use things like auto-inducible media. If we get the media to run out of glucose and give it lactose, then we can get this to happen. And so when we stick this promoter in front of the D7 polymerase gene, now we can then induce the expression of T7 polymerase by adding IPTG or depleting the glucose and adding lactose and getting the protein the T7 to be expressed. Once the T7 is expressed, then if our gene is under the control of the T7 promoter, then our protein can get made. They're often, this T7 promoter is also under the control of the lac operon, in addition to the T7 being under the control of the lac operon. So this is going to be in the bacteria. This is not in your plasmid, but your plasmid is going to have the your T7 promoter, and it often has a T7. It also has a lac repressor as well, a lac promoter as well, so that you don't have unintended, you have a stricter barrier to actually making the protein. And this can be important to prevent like leaky protein expression. So we only want our protein to get made when we add IPTG to induce it. However, sometimes this lac repressor can be kind of leaky. And so if that lac repressor falls off, say, or something like that, a little bit of this T7 can get made. And then if a little bit of the T7 can get made and we don't have a lac repressor here, now a little bit of our protein can get made. If we do have a lac repressor here, however, now both of those kind of have to fall off, and so then it's harder to get your protein made, and this is going to give you a tighter control over expression. This can be important if your gene, say, is toxic to the cells. You want to make sure that you're not making even low levels of it to begin with. If you're really worried about this expression, you can use cells like these p-lyse cells that have this thing called T7 lysozyme, and this is going to inhibit the low levels. It's going to inhibit that T7 polymerase, and so if you have low levels of the T7 polymerase getting through, this is going to prevent those from causing your protein to actually get expressed, and so this is good for toxic proteins. And there's like p-lyse S and p-lyse E. The p-lyse S is going to have lower expression of this lysozyme than the p-lyse E. You can also see, if you look at the various strains, that these expression strains, that there are different things that these have, and you'll see like genotype. For some of these things, they'll have like a delta sign or a minus sign that's indicating that something's missing, as well as various things like that. And often you can just like Google and see what these things actually do, and various things like that. And you also see these will have different antibiotic resistance, which is the next thing I want to talk about. So this is going to be your selection marker. So this is how you know if cells actually, you only let cells that actually have your plasmid survive. So typically what you do is you have on your plasmid, in addition to your gene, you have an antibiotic resistance gene. Then you grow the cells in the presence of the antibiotic, and only cells that have taken in the plasmid will have the antibiotic resistance superpower giver. And so they'll be able to survive, whereas the cells that didn't take in the plasmid won't. Often these antibiotic resistance genes are things for antibiotic resistance against like ampicillin, tetracycline, or kanamycin. So if you see like BLA, that's going to be your ampicillin. I don't remember exactly all of the names for the other ones right at this moment, but you can see that kanamycin, the ampicillin, you might see spectinomycin, which can often be swapped out for streptomycin. And you might see streptomycin, you might see chloramphenicol, various things, various antibiotic resistance things. And it's sometimes you want, if you're using multiple plasmids, then you want to have different selection markers on each of them, so you make sure that they're all, you can grow them in the presence of both of them, and then only the ones that will have both plasmids will be able to survive. So you can see that some of the differences with different pet vectors are in their antibiotic. And so some of them are under AMP and some of them are under CAN. So the ones that I use the most are these 28, pet 28, which are under this CAN. So kanamycin, you grow them with kanamycin. Okay, they might sometimes still have secretion signals. So if they get secreted out of the cell, maybe they'll have signaling peptides that'll get them into like the nucleus or mitochondria or something like that. If it's like for a non-bacterial expression system. So sometimes they might have like secretion signals to make your protein secreted. Then they'll often have epitope tags. So an epitope is this word that we use for a feature that an antibody recognizes. And so if we stick one of these little bit extra, an antibody is just this little protein that's going to recognize parts of something else. And that parts of something else is often a protein, and often it recognizes a little part of a protein, and we call these little parts epitopes. And so if we add a little extra onto the end of our protein, then we can get this like epitope tag. These include like HA, FLAC, MIC, V5, or HIS. And so that brings me to my other point, is that the tags can often also be for purification. So sometimes you might see a HIS tag, you might see a Strep tag. Sometimes they're also like a fusion partner, so that it'll have like a like an MBP or GST on it to help with solubility, as well as potentially with the purification. So when you have a tag that can act as this like affinity tag, now you can have it do things like protein chromatography, where you get that protein to bind to little beads that have a complementary feature to the tag. And then you wash everything else off, and then you can elute or like push off your protein of interest with the absence of all that other stuff. You can also use these tags for detection with things like Western blots, or see what it's bound to with co-IPs. You can do things like, if you're looking in cells, you can have fluorescent markers, various things like this that you might see with these epitope tags. Often these tags will be next to like a protease cleavage site. So this can be like TEV, HRV3C, thrombin, etc. And so you can see that some of the different pet vectors have different tags. And then you'll also see that some of them will be on like the N terminal, and some of them will be on the C terminal. And so some of them, sometimes you'll see vectors that have on both. And so when you see like C or N, that's referring to the N terminus, so the starting end of the protein, or the C terminus, so the ending end of the protein. And know that if you get a plasmid has a tag in one place, you can always just move the tag with like use like PCR to move the tag to the other end, or that sort of thing. So even if the plasmid has it on one end, that doesn't mean that you have to keep it there. So this is, again, with the PCR-based methods like SLIC and things, it's a lot easier to change things around than it was if you had to rely solely on the restriction sites, which I'll get into in a minute. But first of all, note that when we're talking about the protease sites, these are how we're going to cut off the protein, or cut off that tag from the protein if we want to. We don't always want or need to. So this is going to be cutting the protein. But then there are also restriction enzyme cut sites, and this is for cutting the DNA. And this is often for cutting the DNA so that you can then move it into some other place. So in this like restriction cutting method. So restriction enzymes are these enzymes that can, so these like protein reactant mediator speeder upper things. Basically, they're like these ones are like DNA scissors. And so they will cut the DNA when they recognize a specific sequence. And then if you're in your cloning methods, you can then use that to paste it into a different place that has a complementary sequence. So if you say get a plasmid that has these sites on either end, and then you have a vector that has the same sites, then you can cut them both and mix and match. You can also, if your vector doesn't have those sites, you can add them on with PCR and then do this. Or you can just use PCR-based methods from the beginning. But these sites, so you might think, okay, well, how likely is it that they're going to have the same site? But actually, these plasmids are often designed with these multiple cloning sites, or MCS. And so this is going to have a lot of different sequence, a lot of different restriction sites for that, so that you have lots of options. Okay, so let's just look at an example. So this AGO2 was this protein that I studied in my PhD. And you can see that if you look at the vector, so this is the vector, and you can see that it has like all of these different cloning sites. This like MCS, these are all different restriction enzyme cut sites that you can use. And so when you have all of these different options, now this is going to make it easier to clone in what you want. So here's just like a pet vector manual. And I think a key thing is that differences between some of the different pet vectors, like the A, B, C, D, etc., this is going to correspond to differences in the open reading frame with respect to a restriction cut site. And with these different vectors, you're going to have, if you were to use BMH1, you don't have to use BMH1. Maybe you use one that's down here more, and then you'll have more letters in front, but you might have different letters in front if you use one of these different things. But if you were to use this BMH1 site, now what you would see is that here you would be, if you cut here, you're going to be in like a different reading frame than if you cut here, because the ribosome is going to be reading these 3-letter words, and so it's reading them 3, 3, 3, 3, 3. And if you change where the codon starts, then you're going to have different. So you just need to keep this in mind. So when you're looking at your plasmid and what frame it would be in when you cut it, then you would want to make sure that you're in the frame that's compatible with the plasmid that you want to put in, so that you're getting the T7, so that you're getting the ribosome to make the protein in a way that makes sense and not a way that's just gibberish. And so again, this is less of an issue if you're using a PCR-based method like SLIC. Some other thing is that, so another thing to keep in mind is that some bacterial plasmids are actually, you can use this thing called blue-white screening. So selection was when we were just, we were saying, okay, only cells that have this feature can grow. And so with screening, we're saying, okay, well, anything can grow. We're not anything. It still has to have the antibiotic because you're using this in combination with selection typically. But then you want to say, okay, well, show me the ones that have this thing. And so often the thing that we want to see is, okay, well, is our insert in the vector? And so is our instructions, is the gene, is the cDNA actually in the vector like we think it is after we cloned? And so the real way we're going to test this is with sequencing. But first, if we want kind of a quicker idea, we can do things like blue-white screening. And this is also going to use lackey things, but it's going to do a different way. And basically, I have more on this in another post. But if your insert gets in, then it's going to disrupt this gene that's needed to make this product that is going to make a clear or white product turn blue. And so if you interrupt this, if you successfully get your thing into that gene, into that plasmid, then you're going to have white colonies instead of blue colonies. And so, but in order to do this, you have to have a vector that's compatible with this. So your vector has to have that gene that you're going to interrupt. And so some of the plasmids that are compatible with this include like pGEM-T, PUC18, PUC19, and pBluescript. But the host cells have to be compatible too, because they have to make the rest of the protein that's needed. So the part that you're disrupting is really just this alpha peptide, which is just a small part that's going to complement the protein that the bacteria strain makes in order to make the full this full protein that can then make the blue product. And so the host cells need to be compatible too. And so some of the are XL1 blue, DH5 alpha, DH10B, JM109, STBL4, JM110, and top. This is back to the idea that the cells matter and not just the plasmid. So for example, when we have when we're doing our cloning for a protein that we want to express with T7 promoter, initially we stick it into cells that don't even make the T7. So when we're doing the cloning step, we often put these in like DH5 alpha, and then here we might stick it in like a BL21 DE3. So that has a DE3 that has the instructions for making T7. But this way we're not making it until we want it. So it's the plasmid and the host cell that matters. Just a couple final notes before we look again at some examples. So the plasmids also often have a bunch of notation with things like delta something. So the delta means that it's missing. This also can be indicated with like a minus sign. So for example, Rec A, these cells are deficient in an E. coli repair system. And so the system, this rec-based system, is going to carry out this thing called homologous recombination, where basically the bacteria finds DNA sequences that look the same and puts them together. And so this can kind of shuffle things around if it's and so it's kind of like this repair mechanism. So if the bacteria, like if something happens to the DNA and it gets separated, then they can like find things that are the same and use one as a copy for the other, but this can like lead to things getting shuffled around. And so if you don't want this, then Rec A minus cells. But then there are also things that you wouldn't want this for. An NA mutation is another common one you might see. This makes the cells endonuclease 1 deficient. So they don't make this non-specific endonuclease, so like a nucleic acid cutter. So before we talked about restriction enzymes, and those were really specific. These is not specific, and so it can chew up your plasmid. So NA mutation will keep your plasmid from getting degraded. So those are some common things that you might see. An interesting note is that when I was looking and seeing, okay, well, is a pet vector, are these really like the optimal thing? So apparently these were established in like 1987, and then Novogen and Invitrogen and like expanded the series. And these are really commonly used, but this group wanted to know, Patrick Schilling and all, they want to know whether this was like actually optimal and whether you could optimize it. And so apparently they could optimize it. And so in this vector, you can see that you have this lock operator and this T7 promoter. So it's under the control. You have your gene will be here, and it's under the control of this T7 promoter. And then the lack operator is going to repress it in addition to having it repressed or having it not be able to mate if you don't have the T7. Then it has a his tag, a thrombin site, so you can cut off that his tag. And then the multiple cloning site where you can then cut and paste your gene in. It also has an origin of replication. So this is that PBR322 we're talking about. It has a kanamycin resistance gene and this protein for making the lac repressor. So the thing is that the T7 promoter, one of the key things that they realized was that this T7 promoter that's in here, it actually is shorter than the consensus one. So it's shorter than the one that is like usually used in nature. And so they found that if they restored it to its full length, they got better expression of their reporter, which in this case is this super folder GFP. They also wanted to see if they could improve the translation. So that would improve the transcription by having a better T7 promoter, but then the ribosome also has to do a good job at really getting hooked on and making the protein. And so they took this region called this translation initiation region, and this is where it's going to help with coupling it to the translation. And they made a bunch of mutations in it and selected for the ones that were best at making their protein. And this allowed them to find a sequence that was a lot better than the sequence that was already in there, which sequence that was already in there, they say it's likely just because that's when they were cloning things together and it wasn't really much thought given to it initially. Okay, so yeah, you can actually find this plasmid on Addgene. And so you'll see that Addgene gives it this like flaming hot signal, which means that it's really popular. And so when you get it from Addgene, it's often sent, so it's like plasmid sent in bacteria as an agar stab. And so basically you get this little tube and it's got agar, so like bacteria food in media in like this gel, or bacteria food is called media, but it's like in this gel listed all called agar. And then it has like a little like little pipette tip or just a little pool of the bacteria. And then what you do is you're going to streak this out on a plate, typically grow more colonies, maybe make a glycerol stock. So you have a stock for storage, do an overnight growth, so do a mini prep so you get lots of that plasmid. So when you get something, it'll give you a lot of information about it. It'll give you the information about the antibiotic resistance, growth temperature, growth strain, sequencing primers. And then when you use it in your paper, then you cite it, and you give this, say like this was a gift from blah, 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 from, in this case, Daniel Daly. What's interesting is that I didn't know until like grad school, well, I guess late in grad school, that when people say like this was a gift from, it was often because it was from a depository. I was thinking that like, so they're really just like asking, like each time they're just asking this person for their plasmid, but a lot of times it's from actually a gift from a depository. But often you can actually, if something is not in a depository, then there's the option of contacting the authors to see if they'd be willing to share. But so Addgene and DNASU, these are repositories that people that like authors can donate a bit of their plasmid to. The great thing about plasmids is that bacteria will make oodles and oodles of them. But if each lab had to keep like giving it out to anybody who wanted it by themselves, that could be a lot of work, especially if you have a really popular plasmid like this. And so what these repositories do is that they like propagate it, they make a lot of it, they store it, and then people can go through them if they want to get a sample of it. So either on Addgene or, so this is typically where I go first, is Addgene. It's just like, because there's often there's often a lot more like the published ones. There's less, though you might have more luck with like a DNASU if it's something that people haven't studied that much, and they have some great tools for helping you find them. And so, yeah, this is just like a global nonprofit organization, works with different institutions, as well as has a lot of just like sequences that haven't been studied that much. There used to be a plasmid repository at Harvard. There used to also be like this image consortium. Basically, there used to be a lot more than there now is. But thankfully, you can still get most of what you would want from one of these two places. And then, of course, there are other companies that will sell you customly cloned ones. Sometimes you might want to use a custom one if you are doing something where you're going to want to optimize the codons to make it potentially get better expression in a cell type that is not normally made in, or that sort of thing. Okay, so I think that was it. Oh, the other thing I want to mention was that when you look up, when you go, you can also reach resources from like Uniprot if you're looking at a protein, or from GenBank or NS or BLAST or something. There'll often be like links to different sources, commercial sources where you can get things. If you have, when you search for, say, DNASU, what you're going to see is, so you can see that there are a lot of different ones, and there are a lot of ones even that say that they're in the same plasmid. So you have 3 of these in the same plasmid. You also see that there's like closed and fusion. So fusion, it doesn't have a stop codon in the way that it's cloned in. So this is just for, these are just for cloning, and for not for expression. If you were to try to make it express, there's no stop codon, and so it wouldn't work. You get some like weird run-on protein. But when closed, it does have a stop codon. But these, you can see these are different clones for the same protein. Often these have different, these will have slightly different like sequences, or they can have different, sometimes there are alternative variants. So see like short variant or partial clone comparing to other ones. And so you want to make sure that if that you're getting the variant, or maybe it's like a splicing variant or various processing variant. Maybe there's an alternative start site. So you want to make sure that you're actually getting the one that you want. But then remember that it's okay if it doesn't come in the vector that you want, because you can then subclone it into the vector that you do want. And hopefully now you know a little more about how you might know what you would want to clone it into. So hope that helped.
Video này do mình, cương bên npslab.vn, tự chỉnh sửa phụ đề hoặc lồng tiếng bằng công cụ, thấy vậy thôi, chứ tốn công lắm á. Thấy hay thì nhớ bấm theo dõi kênh, cho mình thêm động lực làm mấy video phòng lab xịn sò nữa nha.